In today's video, we are going to be talking about cellular respiration and looking at the glycolysis reaction in some detail. So there are two types of cellular respiration: aerobic, that is with oxygen, and anaerobic, that's without oxygen. And the primary reactant for both of these is glucose. And so the same first reaction is used by both aerobic and anaerobic. And this is glycolysis, where we convert glucose to pyruvate. So simply put, this is the breakdown of glucose into two pyruvates. Glucose is a 6-carbon molecule, whereas pyruvate is a 3-carbon molecule. And there are 4 basic reactions involved in the glycolysis reaction. We have phosphorylation, lysis, oxidation, and substrate level phosphorylation. During this process, there are a total of 4 molecules of ATP made per glucose, but there are also 2 molecules of ATP utilized during this reaction, and this will give us a net gain of 2 ATP per glucose molecule. So the first reaction, phosphorylation. So this reaction adds phosphate groups to our glucose, and there will be a total of 2 phosphate groups added, and these phosphate groups are contributed by the breakdown of 2 ATPs into 2 ADPs, and our resulting molecule is a fructose 1,6-bisphosphate. So still 6 carbons, but now with 2 additional phosphate groups. The second process is lysis. Now lysis means to split. So our fructose 1,6-bisphosphate is split and creates 2 3-carbon molecules. Now one of the common names for these molecules is just triose, which just means 3 carbons, sugar. But they can also be called phosphoglyceraldehyde, which is PGAL for short, and I tend to use PGAL. So throughout this we will use the abbreviation PGAL. So we get 2 PGALs per fructose 1,6-bisphosphate. Then we go on to oxidation. Now both PGAL molecules will be oxidized separately. So from this point we're just going to follow one PGAL, but assume that everything happens to both PGALs equally. So again, prior to oxidation, we're going to need to add an additional phosphate group. Where this phosphate group comes from, we're not going to worry about that detail. But we know that if something is being oxidized, then another molecule must be reduced in the process. And here, for this process, we're going to use the hydrogen carrier, NAD+. When this is reduced, it becomes NADH plus H+, and it carries away hydrogens and high energy electrons. So our PGAL is oxidized, and in the process our NAD+ is reduced. The final form after oxidation is a 1,3-bisphosphoglycerate. So now 2 phosphates on the first and the third carbon, and still 3 carbons in total. And our NADH plus H+ will come into play later on. The final part is substrate level phosphorylation. This is literally the reaction that creates ATP at this level. Now there are 2 phosphates on our carbon compound, and so this allows us to make 2 ATP from that compound. So it uses 2 ADP and takes the phosphates from our 1,3-bisphosphoglycerate to form 2 new ATP. And the final product of this reaction is pyruvate, our 3-carbon molecule we want. So our 1,3-bisphosphoglycerate gets the phosphates removed, the 2 ATP become 2 ATP, and we get a pyruvate. And of course this occurs twice, so we get 4 ATP and 2 pyruvate molecules. So a review of the glycolysis reaction. It's the first set of reactions in all cellular respiration processes, whether it's anaerobic or aerobic. It uses 2 ATP but makes 4 ATP, so has a net gain of 2 ATP. There are 4 different reactions involved: phosphorylation, the addition of phosphate groups; lysis, splitting of the molecule; oxidation, which is the removal, in this case, of high energy electrons and hydrogens; and substrate level phosphorylation, the creation of our ATP molecules. Our end products that go into our next step are 2 pyruvates, 2 NADHs, and 4 ATP. Now, the video on anaerobic respiration will tell you how our pyruvates and our NADHs are utilized in that process. And our next video will look at how our NADH and our 2 pyruvates are utilized in aerobic respiration. Any questions? Just put them in the comments, and good luck.